So it's been a break. Let's catch, where were we? We were talking about the liver and the pancreas before, and now we're going to talk about the liver some more and intestines. So we're working our way down the digestive tract. And so we're going to review some liver anatomy, and then we're going to talk about blood flow. So blood flow in the digestive system is complicated. And because it combines your knowledge about the cardiovascular system and the digestive system, you can imagine that I might be attracted to asking you a question about blood flow and control of blood flow in the digestive tract. I'm just giving you a heads up that this is coupling all the way back to the beginning of the course, and I like to tie things up like this. So we'll talk about blood flow and how complicated it is, and then we'll talk about liver anatomy and physiology, and then we'll get into intestinal anatomy and physiology. I think I showed you this slide before break. These are the most important functions of the liver. The one we're going to be focusing on today is the secretion of bile salts for the digestion of fat and bicarbonate for the neutralization of gastric acid. We talked about that a little bit before break. Hopefully you thought about that a little bit while you were eating and digesting your holiday meals. But there are some other functions, and I would not ask you to list 8, but I might ask you to come up with 3 or 4 other functions besides the ones that we focus on today, like synthesis of angiotensinogen, or storage of glucose as glycogen, or storage of protein as fats, fatty acids, that sort of thing. Remember, this is an anterior view of the liver. Here's a cartoon. We have, the liver has ducts that we'll talk about some more in a few minutes. The main bile ducts, the left and the right bile duct, join to form the common hepatic duct. The common hepatic duct joins with the cystic duct, which is not labeled here, but is one of the things on your list. You might want to decorate your slide with a label here for the cystic duct. The cystic duct and the common hepatic duct join to form the common bile duct, which empties into the duodenum. The empty point is called the sphincter of Oddi, or the ampulla of Vater, or a number of other names. The pancreas has a pancreatic duct, and main and an auxiliary pancreatic duct that also delivers pancreatic juice to the duodenum. So these are some of the plumbing terms to know about. This is not blood flow. This is bile flow. Well, this is bile flow. This is pancreatic juice flow. So here's a drawing from an old anatomy text. I use it because it's pretty descriptive. Now we're going to talk about blood flow to some of the digestive tract. And these are important terms to know. So let's get oriented. Here's the stomach. Here's the pylorus. The pyloric sphincter is behind it. The liver has been sectioned away, so the liver is missing, right? Big section of the liver is missing from this image. Here's the gallbladder. So right there is the cystic duct. There's the common hepatic duct, and there's the common bile duct. Okay, let's go back to blood flow. Here is the abdominal aorta. Branching out from the abdominal aorta is an artery, a major artery called the celiac trunk. We did not see this tube on the cadaver, but we've seen its hole. We cut it right here, and so the hole in the abdominal aorta is visible. And so if I pinned that hole, you would say, well, you could ask me, are you asking for the artery that's coming out of this, or are you asking for this big tube? And I would say, I'm asking for the artery that's coming out of here that's cut, that is in this hole. And during the practical, just as a note, you're allowed to ask me questions. You're not allowed to ask me questions that use an anatomical term, right? So you can't say, are you looking for aorta here or celiac trunk? I mean, you can't ask me that because that gives the answer away to everybody else in the room. If you need to use anatomical terms, we can step out of the cadaver lab and you can ask me those terms, use those terms. But if you're in the lab, you can say, are you looking for this big thing or are you looking for this little thing, right? That doesn't give anything away, and I will answer those questions. Okay, so the celiac trunk comes out of the abdominal aorta and then it branches. It does a three-way branch. So we haven't seen this on the cadaver, so I couldn't pin this on the cadaver, but I could ask you about this on a paper quiz that shows you a cartoonized version of it. This three-way branch is the left gastric artery. That's the left gastric artery. The splenic artery, which on this cartoon, because it's so old, it uses an archaic term for the splenic artery called the leonal artery. I'll accept both, but these days it's called the splenic artery. So we have the, the left gastric? Yeah. Left gastric, splenic, and the common hepatic. These are all arteries branching off of the celiac trunk, which is a tube that comes out of the abdominal aorta. Okay, so we've identified these three major arteries that branch off of the celiac trunk. Now let's follow them one at a time. The left gastric goes around the lesser curvature of the stomach. And it's going to join into the right gastric. So we have the left gastric and the right gastric are joined end to end. The end of the right gastric and the end of the left gastric artery are joined. The right gastric is a branch off of the common hepatic artery. So let's follow the left gastric. The left gastric comes off of the celiac trunk and then vascularizes the lesser curvature of the stomach. The common hepatic has a branch that branches into the right. One of the branches from the common hepatic is the right gastric that also vascularizes the lesser curvature. Okay, so we've done the gastric artery, left and right. All of the veins, right, so it's unusual for arteries to connect to arteries. They usually connect to veins. These arteries all connect to veins down in here. Right, so small arteries, arterioles, venules, etc., capillaries, venules, etc., all of that stuff is down here in the stomach itself. There's a name for this thing where arteries connect to arteries, and I'm blanking. I think it's anastomosis or something like that, but obviously I'll have to test you on that. It's not the only place where arteries connect to arteries. Okay, so let's follow. So this splenic artery goes to the spleen, and it also provides blood flow to the pancreas. So the splenic artery provides blood flow to the pancreas and the spleen. Okay, so we've done this one and we've done this one. Now the hepatic artery, well, one of it comes off of here doing the right gastric. And then there's one here, and you'll see that we're going to cover that in the next slide. But a major branch of the common hepatic is the proper hepatic artery, or just the hepatic artery that goes to the liver, as the name would suggest. Okay, so I think those are the main arteries that we want to know about here, coming off of the celiac trunk. The next slide is strange because this is another cartoon from the same book. By the way, it's called Gray's Anatomy, so that this is a classic book of anatomy from which the TV show got its name. Right? The book came before the TV show. So in this case, the stomach has been flipped up. So this is the lesser curvature of the stomach, and this is the greater curvature of the stomach up here. The stomach has been flipped upside down in this image. Anatomists are allowed to do that. So here is the celiac trunk. Here is the splenic artery. You can see providing blood flow to the pancreas and to the spleen. Here is the, let's see, here's the left gastric, providing blood flow to the left side of the lesser curvature of the stomach. And here's the right gastric coming off of the hepatic. So this is the same stuff that we saw before. Now let's look at what else we have. Branching off of the splenic, providing blood to the greater curvature of the stomach, is the left gastroepiploic. That comes off of the spleen. This one, which you could predict is the right gastroepiploic, branches off of the hepatic artery here. Actually, this part of it is called the gastroduodenal. So coming off of the hepatic is the, what did I tell you about before? I told you about the right gastric. Now I'm talking about the gastroduodenal. And from that gastroduodenal right there, right here, there's a branch to the right gastroepiploic. Greater curvature of the stomach. So I could ask you a question, something like, what arteries provide, blood is coming from the aorta and it's destined to go to the greater curvature of the stomach. What arteries does it go through? Well, you'd have to be thinking about, celiac trunk to the splenic, to the left gastroepiploic, and you'd have to be thinking about celiac trunk to the hepatic to the right gastroepiploic. Oh, and the gastroduodenal as well. That's how you would answer the question, to follow blood flow from the aorta to the greater curvature. That's a hard question. The right gastric on this is right here. I know it's hard. This is some of the harder anatomy, and that's why we're doing it in this room and not only in the cadaver lab, because it's hard to see in the cadaver. Questions about this? So I'm trying to make this sort of like an outline. Celiac trunk gives rise to these three. Splenic gives rise to this. Hepatic gives rise to this, these two. And gastroduodenal gives rise to the right gastroepiploic, as well as arteries that feed the duodenum, as the name would suggest. So gastroduodenal, right gastroepiploic, and duodenal arteries. And they have names: superior, inferior, medial, blah, blah, blah. Just, we're not going to worry about those. Duodenal, providing blood flow to the duodenum. Questions about this one, and about this one. It's the same material, it's just different ways of imaging it. So can we see the gastroepiploic artery here? So yeah, here is the right gastroepiploic that was connected to the gastroduodenal, which is behind it. Maybe this might be, I suppose this is the gastroduodenal. Then behind the stomach, it branches and forms blood flow to the right gastroepiploic. And behind here, here's the spleen. Behind here, coming off of the splenic, is the left gastroepiploic. Both of those, right and left gastroepiploic, providing blood to the greater curvature of the stomach. The only ones that we would see on the lab, in the cadaver lab this week, the only one of any of these labeled here would be the celiac trunk. So for the cadaver lab, for the practical, that's the only one to know. But for the final, all of this. Okay? Okay, that's blood flow to the organs, to the liver, to the stomach, to the duodenum. Now we have to talk about venous blood flow. And the most important one that we're going to talk about right now is the portal vein. Right here, the portal vein. This is a vein that brings blood from the stomach and the intestines to the liver. This is the vein that brings nutrients from your digestive tract to your liver for processing. So we haven't talked about how your intestines get those nutrients yet, but because we're still talking about liver, but that's why we're going to talk about this. So the liver gets two kinds of blood. It gets arterial blood from the hepatic artery, and it gets venous blood from the portal vein. That's what we're focusing on right now. We'll get into these details late in a few minutes, because we're still focusing on liver a little bit. So gastric veins, gastroepiploic veins, splenic veins all drain into the portal vein, which takes venous blood to the liver. Okay, so let's see how the liver is built. So we're focusing on the liver again. The liver is formed from these, so here's the liver. Here's some names. So there's a hepatic artery. We talked about that's arterial blood being brought to the liver. And here's the portal vein, bringing venous blood to the liver. The vena cava is going past, it's going through-ish the liver. It's not bringing, the inferior vena cava is bringing blood flow back to the heart, not to the liver, but it's going right next to the liver. So there's a tube that we saw in the cadaver that is the inferior vena cava that I could label because it's right there with the liver. But it's not bringing venous blood to the liver, it's bringing venous blood past the liver to the heart. Okay, so if we dissect out a chunk of the liver, it has a stereotype structure called a lobule. And in certain staining, it actually shows up as this hexagonal array. So each of these lobules has blood flow, both arterial and venous blood flow. And so here's a blow-up of part of a lobule. And in the center is an actual vein. So here is blood, here is arterial blood being brought to the cells of the liver. Why does the liver need arterial blood? Well, because it's an active organ and it needs arterial blood with oxygen to provide for oxidative phosphorylation and all the energy needs of the liver cell. So it needs arterial blood just like any other cell in the body, but it also needs venous blood that is rich in the nutrients that it's trying to process. So here's the arterial blood and here's the venous blood. Now where arteries and veins are usually connected through capillaries, the liver is an exception. There are no capillaries between the arteries, the arterial blood, and the capillaries. Instead, there are spaces between the hepatocytes, between the cells of the liver, called sinusoids. They're just spaces. It's not tubes. So blood flow is sort of percolating through around the cells of the liver. And arterial blood and venous blood just sort of work their way out, surround the cells through these sinusoids, bathing the hepatocytes in a mixture of arterial and venous blood. Percolate, that's a word. So, when water drains into ground, and so if it doesn't flow off the surface, if it sort of drains into the ground and the ground soaks it up, that's percolation. When you pour water over coffee grounds and make coffee, that's percolation. The water is percolating around, through and around the bits of coffee, of ground coffee bean. That's what percolation is, and that's the term I'm using here. The blood is sort of working its way around the hepatocytes. Like capillaries, but not capillaries. Sinusoids. Okay, so the sinusoids are bathing each of these hepatocytes, they're cuboidal in shape, with arterial and venous blood. Remember, the veins are bringing the nutrients to the liver for processing. Storage of carbohydrate as glycogen, storage of fats as fatty acids and triglycerides, storage of protein as triglycerides. Yes?
Where did you say that there were no capillaries?
There are no capillaries in the liver proper. There's no capillaries in the liver proper, inside the body of the liver. It's sinusoids. The other tube in the liver are bile ducts. Remember, one of the things that liver does is make bile. It takes energy, that's why we need arterial blood. It takes energy to produce bile. And then the bile collects into these bile ducts, little tiny bile ducts, which then drain into the left and the right hepatic duct, which drains into the common bile duct, well, which mixes with the cystic duct to make the common bile duct, right? So these microscopic ducts are going to join in to form, let's just, these small bile ducts are going to join together to form a major duct, like the left or the right hepatic duct. So bile, which is produced by the hepatocytes, is excreted into these little tubes, which are normally green. I mean, they are, I mean, this cartoon is not really an exaggeration, because bile is quite green in color, and so these little tubes are greenish in color. So here's an image. This is not a cartoon. This is an actual micrograph of hepatic lobules. So here is the central vein. So we have right here, this would be a branch of the hepatic duct. A branch of the portal vein, a branch of the bile duct, and a branch of the hepatic artery. Blood flow from the portal vein and the hepatic artery are percolating around the hepatocytes. These white spaces are the sinusoids that blood is moving through on its way, bathing all of these cells on its way to the central vein. Let me go back one. So all of this percolation, arterial blood and venous blood, then works its way to a central vein, which is going to become the hepatic vein, which is going to drain the blood from the liver into the inferior vena cava. Yeah.
So the central veins are branches of the hepatic veins?
Yes, are sort of precursor. So yes, coming off of the hepatic vein are these many central veins. Each lobule has a central vein. Remember, this is microscopic. Yeah.
In regards to where you had just said that was the hepatic artery, where that was cut, it looks very, very similar to the bile conductuals, like at least that, the one, yeah, that one. You said that was a hepatic artery, right, originally?
So I'm not sure, I can't really tell the difference between these two. I can tell that this is a vein. So this is the branch of the portal vein. One of these is an arterial branching from the hepatic artery, and one of these is a ductual that is going to join into form the hepatic duct. I can't tell which is which. So you can't tell which is which. Well, actually now I can, because this is labeled. So if these are bile ductules, then so is this. And if this is an arterial, then so is this. Right? And this is a branch of a venule from the portal vein. Okay? So this is what's providing venous blood that's going to percolate through this lobule. This is what's providing arterial blood that's going to percolate through this lobule. And this is what's going to drain bile from little tiny vessels here into a less tiny vessel that will join into the bile duct. Does that help? The white spaces are the sinusoids, analogous to capillaries, but not capillaries, because they're not lined with endothelium. Okay, so the liver produces bile. Bile flows into the bile duct. So here's the right hepatic duct, here's the left hepatic duct, here's the common hepatic duct, joins with the cystic duct to form the common bile duct, all these names are, none of these would be labeled on the cadaver, none of these are for the practical quiz except for the gallbladder. I could label the gallbladder. So the liver is producing bile. The bile can go straight into the duodenum for digestion, but in between meals, the sphincter of Oddi is closed, and the bile that is produced by the liver backs up and fills the gallbladder. And then during a meal, the gallbladder squeezes. It's one of the responses to those hormones that we talked about last time, and we'll talk again about. Cholecystokinin, that hormone that is triggered when chyme enters the duodenum. Cholecystokinin triggers the contraction of the gallbladder and the relaxation of the sphincter of Oddi during the digestive phase, and triggers squeezing of bile into chyme to help with the digestion of fat. Trust me, I know this is hard. It's taken me years to learn, and you don't have years. Okay? So let's talk about, so we've talked about blood flow to the liver, right? We've talked about bile flow from the liver. Now we're going to talk about what bile does. Bile is a derivative of cholesterol. It's a detergent-like molecule. It's got a non-polar side and a polar side. It's amphipathic. It acts like a detergent. It solubilizes fat. So now let's talk, and that's what it's for, the digestion of fat. So let's talk about the digestion of fat. So we're transitioning from anatomy to physiology now, talking about the digestion of fat. Here's a glob of fat. You've eaten a piece of buttered toast. The carbohydrate is being digested by amylases. Now we have to think about the butter part of the toast, or the baked potato that you've put butter on. Okay? The fat forms a small glob, right? It's not soluble in water. It's not soluble in acid in the stomach. But we need to solubilize it if we're going to get the goodies, the nutrients that we want from fat. Fat is it's got a lot of energy content. It's good stuff as far as providing energy. So the fat globule is emulsified in the stomach. All that churning, remember the stomach with all that, churning action, that turns the fat glob into smaller bits, combines with the bile salt to emulsify. So so you've seen an emulsion. If you take oil and water and you shake it up, just take a jar of oil and water. Oil will be on top, water will be on the bottom. You shake it up, you put it on the table, what happens? The oil goes to the top, the water goes to the bottom. They're immiscible, they don't emulsify. But if you take some oil and you take some water, let's just say it's slightly acidic water, maybe vinegar, and you take some egg yolk, which is an emulsifier, and you shake it up, you get mayonnaise. Mayonnaise is an emulsion of fat, water, vinegar, and egg yolk. Look at the ingredients on a jar of mayonnaise. You'll see that the main ingredients are oil, vinegar, and egg. Well, we don't have vinegar, but we have gastric acid. We don't have egg yolk. We have bile as an emulsifier. So its first job is to turn this glob of fat, which ordinarily would just separate from water, and make it small enough so that it's now in an emulsion of tiny fat droplets in aqueous solution. It's not really a solution, it's an emulsion. We're clear about what an emulsion is? Mayonnaise is a perfect example of the kind of emulsion we're talking about here. So this emulsion of fat and phospholipid from bile and bile salt from bile forms these tiny droplets. So here is the glob of fat that was eaten. Here's the emulsion droplet that is formed in the small intestine, in the duodenum, when it's mixed and churned up with bile. These emulsions form micelles. How many of you know from biochemistry maybe, what a micelle is? Okay, some of you. Provide a definition from biochem. Somebody. Anybody remember what a definition of micelle is? So the one tail that we're talking about here is the bile salt. So the micelle, the micelles that we're talking about here are mixed macromolecular assemblies of phospholipid from the fat that we eat, bile salt. I'm just going to draw the bile salt as a blob here. Bile salt. And, well, let's just call it like that. So but there's no, this is not a vesicle, right? The difference between a vesicle is that the bilayer has an aqueous interior. This is a mixed micelle of lipid, detergent, and it maybe have some hydrophobic proteins mixed in there too. It would be any fat-soluble protein might be in this micelle. We're focusing on protein, not on, we're focusing on fat, not on protein. So what we're doing is we're working our ways tinier and tinier. So the fat droplet is a tiny little droplet of fat. It is emulsified into smaller droplets of fat. This emulsion droplets are solubilized, actually dissolved to form micelles, which are these populations of lipid, fat, and detergent. And from those micelles, we can get monomers of fats. Monoglycerides, maybe some triglycerides, some fat from, remember lipase. Lipase now has lots of surface area to work on the lipids, to cut up the lipids into fatty acids. This is what we need to absorb. We can't absorb droplets of fat, we can't absorb micelles, and we can't absorb fat droplets, we can't absorb emulsion droplets, we can't absorb micelles, we absorb monomers of fats. We got those monomers by breaking the fat up into smaller and smaller bits, giving the lipase, remember we have lipase from the pancreatic juice, an opportunity to cut the fat into these monomers. These monomers are amphipathic, and so they simply diffuse into the cells of the small intestine. So now we're talking about how we absorb the nutrients from fat. Diffusion, this is the apical membrane, here's the basolateral membrane of a epithelial cell surrounding the intestine. Diffusion into the cell then assembles, now this is important, it assembles these fatty bits into something called a chylomicron. Where's the name? Chylomicron. These are chylomicrons. It's an assembly of triglycerides and lipids. Fat, triglyceride and lipid. Chylomicron. It's also got LDL, low-density lipoprotein, and HDL, high-density lipoprotein. We've heard of these when we think about serum cholesterol. In any case, the intestinal cell assembles the monomers into chylomicrons and then exocytoses them. You know all about exocytosis from cell-phys. Exocytosis of the chylomicrons is then picked up not by blood, here's the twist, not by blood, but by a branch of the lymphatic system called the lacteal. So where is this from? So let's just skip this. Here is a image, this is the lumen of the intestine. Here is an intestinal fold, you've seen that on the cadaver. Each of these cells has microvilli, that's illustrated here. Each cell has microvilli. The intestinal cells are what assemble the fat that dissolved into it, into chylomicrons. Those chylomicrons were exocytosed into this space, and then they were picked up by this lacteal, which joins into the lymphatic system. So fat goes into lymph first, and then it goes into blood. the lymphatic system drains into venous blood in the left subclavian vein. It's a little weird. All the other nutrients go straight into blood. Okay, let's take a break before we start talking about absorption of other things. What we've done now is we've talked about the absorption of fat. Now we're going to talk about the intestinal absorption of sugar and the intestinal absorption of protein. That's what's coming up. Let's take a break. The heavy lifting has already been done. The rest of this is pretty straightforward. Well, the physiology is straightforward. We're going to do some anatomy review, which is not straightforward. Right? So let's do the physiology, which is easy by comparison. So what do we have here? Lumen of the intestine. This is the small lumen of the small intestine. Here is the blood side, apical membrane, basolateral membrane. Right? Just getting oriented. Here is an intestinal cell that has all that machinery for absorbing fat. Fat is absorbed, then assembled into chylomicrons and exocytosed out here. All of that's happening here too, but we're not looking at that now. We're looking at the absorption of sugar.
And do we have to know that eventually? Like
Know
the
what?
fat, like how the fat like enters and gets transferred into chylomicrons?
The machinery associated with the assembly of chylomicrons is not something that I'm teaching and you don't have to know. It's complicated. Okay? So all you do have to know that the fats, the fatty acids, diffuse into the cell and then black box, magic happens, chylomicrons get assembled. That's one of the few cases where a molecular mechanism is not something I'm expecting you to know. It's one of the few cases, so enjoy it.
Yes.
Now we're going to talk about molecular mechanisms that I do expect you to know because these are easy. We're talking about the absorption of sugar, glucose, fructose. So remember that carbohydrate was broken down by amylase. Remember, amylase is the enzyme that breaks down carbohydrate, and it's both salivary in origin and pancreatic in origin. Salivary amylase and pancreatic lipase broke the starch, the carbohydrate, down into sugars. Here are the sugars. Well, no, these are not sugars. Maltose, isomaltose, sucrose, and lactose are sugars. They are disaccharides. So the table sugar, sucrose, is a disaccharide. Maltose, which is another sugar that is not table sugar, but is used in baking, is a disaccharide. I don't expect you to know which sugars are in the disaccharides. I can tell you if you're interested, but I don't expect you to know that. I do want you to know that sucrose and lactose are disaccharides. Sucrose is table sugar. Lactose is milk sugar. Milk is sweet-ish. In your intestines, in the lumen of the intestine, there are enzymes that break the disaccharides into monosaccharides. So maltase breaks maltose into glucose. Sucrase breaks sucrose, and I forget the two sugars, glucose and one other. Glucose and fructose. Glucose and fructose. Lactase breaks lactose into glucose and galactose. Okay, so I don't want you to know which ones are which, but the sugars we're talking about are glucose, fructose, and galactose. Those are the monosaccharides that come from maltose, sucrose, and lactose. those disaccharides are broken down by these enzymes, and thankfully the names are helpful. Maltase breaks down maltose. Sucrase breaks down sucrose. Names are helpful. The monosaccharides are absorbed in the, so let's talk about glucose and galactose. Here's a protein you've seen before in the kidney, the sodium glucose co-transporter. Galactose and glucose are carried by the sodium glucose co-transporter from the lumen into the cell. They are not just carried, they are pumped using the sodium gradient. Sodium gradient came from the sodium potassium ATPase, just like in fermented every other cell. So this is a story you've seen a number of times before. So I could ask you, what is similar about the intestinal epithelial cell and a proximal tubule cell in the kidney? Well, they both have sodium potassium ATPases. They both have sodium glucose co-transporters. There's probably other similarities, but those are the two most obvious ones. They both have apical membranes and basolateral membranes. There's another obvious one. Anyway, I could ask you to pull this together as part of the comprehensiveness of the final exam. Anyway, here in the intestine, sodium glucose co-transporter pumps glucose and galactose into the cell, and then the glucose uniporter allows glucose to flow down its concentration gradient and get picked up by a blood capillary. This story is just like the proximal tubule. Fructose is the weird cousin. Fructose does not have a sodium-dependent co-transporter on the apical membrane. It simply has a uniporter on both the apical and the basolateral membrane. So fructose flows down its concentration gradient passively, whereas glucose and galactose are pumped and then flow down their concentration gradient. So that part is, you've seen before, you've already learned it. Questions about that? This is the molecular mechanism for the absorption of sugars from carbohydrate. Amylase, these enzymes, absorption of the monosaccharides. Questions about this? Yeah.
What happens to galactose when it's pumped into the epithelium?
Galactose and glucose can both be carried by the glucose glucose uniporter.
Into the interstitial space?
Yeah. So this is a good option. So some of you are probably don't produce lactase anymore. Humans evolved to produce lactase during infancy when we were drinking milk, but evolutionarily, humans stopped producing lactase when milk was not a food product anymore. In evolutionary times, we stopped producing lactase because we were no longer consuming lactose. But the development of agriculture, the development of cows as a source of milk, gave a competitive advantage to human beings who could continue to produce lactase and continue to digest milk sugar. So in the population now, many humans continue to produce lactase in adulthood, and some don't. So some of you probably avoid eating milk or cheese products because it makes you feel bad, because you don't have the enzyme necessary to break lactose down into glucose and galactose that can be absorbed. And so what happens is if you drink milk and you don't have lactase, the lactose stays in the intestine bloating, diarrhea, unpleasantness. So it's called lactase deficiency because white people named it. White people typically have lactase. So, but it's the Northern Europeans, the people who evolved to that, where this mutation of continued production of lactase was beneficial, that are the mutants. The wild type human being shut down the production of lactase because the wild type human being stopped being exposed to milk after infancy. So those of you who have been told you're lactase deficient, you can say, no, no, no. I'm lactose intolerant, but I'm not deficient in any way. It's you are the mutants that are overproducing lactase when you shouldn't be. Shouldn't, that's a judgment. So that's the evolution of lactase. That's a real short story of the evolution of lactase. So the persistence of lactase is about 10,000 years old. It's not very old. Okay, so now let's talk about the digestion of protein. This looks really complicated, but I'm going to make it simple. Remember that proteins were broken down into polypeptides, oligopeptides, by gastric pepsin and by peptidases. What were some of the peptidases that came from the pancreas? Trypsin, chymotrypsin, carboxypeptidase, and those came as zymogens. Remember the zymogen story? So we had procarboxypeptidase, and we had trypsinogen, and we had chymotrypsinogen that got turned into trypsin, chymotrypsin, and carboxypeptidase. Those are all lumped into this one word, peptidases. You know better than to lump them into peptidases because that doesn't get you full credit. If I ask you what are the enzymes that break down protein, you would tell me pepsin, that came from pepsinogen. Those amino acids, so oligopeptides are broken down into amino acids by these enzymes as well. Now there's a complicated set of machinery that we can absorb some tripeptides and some dipeptides, but let's not worry about it. The point is there are sodium amino acid co-transport. This is a sodium-driven amino acid transport, and then amino acid uniports out the basolateral membrane. So this looks just like carbohydrate metabolism, except that we've got all this dongle stuff here about breaking down peptides, and let's not worry about that. The main point is right here. This is the main point of this thing. Sodium gradient came from the sodium potassium ATPase, pumps amino acids into the cell, and then they flow down their concentration gradient through a amino acid transporter on the basolateral membrane into capillary blood. Questions about protein. So we've just done absorption. We've talked about fat before the break, and now we've talked about absorption of starch, carbohydrate, and now protein. This is how these nutrients get into your blood. Where do they go? Well, in the case of fat, they go into lymph. In the case of carbohydrate and protein, they go into blood, venous blood, portal vein, liver. Let's talk about some anatomy. Let's do this game. Unfortunately, my laser that I want to pass around has died. So I'm going to pass around this one. Who wants it first? Abby, so you can first. So, this is the laser button. Let's start first. Okay? So pick one and give me a name. I didn't see what you did.
Oh, I pointed for this line here.
Yeah?
That's the
Yeah, good.
Yeah, I mean, that's points on a test, so.
But I could say, what part of the stomach is that actually pointing
Okay,
to?
go back down.
No, that's okay, just tell me. Oh,
Oh, I'm sorry.
It's the star button,
Okay.
sorry.
Wait.
Go forward, there we
Okay,
go.
there we go.
What
Sorry.
part of the stomach is that line, is the line that Abby pointed to actually pointed to?
Oh, the body.
The body would be an acceptable name, or you could say that's the lesser curvature, depending on where, so both, I mean, it's not exactly pointed to curvature, but I would accept it. Okay, go ahead.
This little thing right here is the duodenum.
Yes, it is.
Okay. Is the next part the jejunum?
Yes, the jejunum is behind, is after the duodenum. The starburst thingy is the reason.
Appendix.
That's the appendix. That little dongle is the vermiform appendix.
Is that the cecum?
Yeah, so that's the cecum right there. This right here is the cecum. Brandon, the laser's coming to you.
This is the mesentery.
Yep, the yellow stuff that we cut out of the cadaver in order to elongate the intestines is the mesentery.
Duodenal jejunal flexion? Or actually, that'd probably be right here, right?
Yes, so that's not this is not a name that I would expect you to, I didn't teach you that. It's not on the key concepts list. The duodenal jejunal flexure would be the joint between the duodenum and the jejunum, so right there. I would not test you on that.
Oh, wait.
But I would test you on ileocecal, where, Matt, point to the ileocecal junction.
That would be right here.
Show me again. This is the
Oh, sorry,
ileum.
I didn't do that yet.
This is the ileocecal junction, the joint from the ileum into the cecum. I think those are all the important ones here. Ascending colon, right? Cecum and then ascending colon. Let's do some more. Now we're going to be talking about some blood flow. Well, maybe. Yeah, we'll see.
Ascending colon.
Good. Okay? Okay?
Yeah.
Let's do the ones that are gonna be important. Which one is the ileum? So, Shay, I'm gonna ask you to point to the ileum.
This one?
No, those are both pointing to the large intestine. The ileum is a section of the small intestine. This is the ileum right here. Ileum, ileocecal junction. This is the descending colon. Up there is the transverse colon. It's got some additional structures called the tinea coli and the epiploic appendages that are not on your key concepts list. But the one of the arteries I do want you to know, so we're getting back to blood flow again, is the superior mesenteric artery. We have seen on the cadaver the whole of the superior mesenteric artery coming out of the aorta. It's not shown here, But this right here is the superior mesenteric artery, providing arterial blood to the intestines. This here is the inferior mesenteric artery, which is not on your key concepts list, I don't think. So I do want you to know that the superior mesenteric artery provides arterial blood to the intestines. And now we're going to look at some veins, I think. Well, okay, well, let's do this. So this is artery. Remember we did, at the beginning of this class, we did all of that complicated arterial blood flow to the liver and to the stomach. Now we're going to do arterial blood flow to the intestines. But I don't want you to know all of these. What is this? What's this tube right here? That's the aorta. What's this tube here? Branching, when the aorta branches into its two main branches at the bottom of the aorta, abdominal aorta. Iliac. The left and the right common iliac arteries. You knew those once upon a time. Because it was on the list for the cardiovascular system. This is the superior mesenteric artery. And it branches into a bunch of colic arteries. So I might ask you, if I pointed to something like that, and you just told me colic, I would say, okay. If I point to something like this, and you say colic, I would say not okay. That's the superior mesenteric artery. If I point to this, and you say colic, you're way off. That's the abdominal aorta.
What is the second one coming off
This one here,
of? Yeah.
the inferior mesenteric.
Oh.
We did not see the inferior mesenteric on the cadaver, but I think it is on your key concepts list. I'll have to double check that. I know the superior mesenteric artery is. If I do this, let's see. So here is the key. Here is the key to those arteries, right? One, can't see them both at the same time. 1, 4, and 6. 1, 4, and 6 are all pointing to the superior mesenteric artery. (13) is pointing to the inferior mesenteric artery. That one. There's the abdominal slit into the left and the right common iliac. Right? Those are the ones that I think are fair game. There's a bunch of others. If you want to show off, you can learn the names of these other others. That's arterial blood to the intestines. Blood has to come from the intestines. Where are we going? We're going to the portal vein. Who's got the laser? I can't reach that anymore. So here is the portal vein, number 16. Portal vein. What do you, knowing what you know about the names of the arteries that are here, what do you think are the names of these veins? Gastric. Knowing what you know about the names of the arteries that are here, what is the name of this vein? The gastroepiploic. 6. Gastroepiploic. So gastric vein, gastroepiploic vein, colic veins. What is this one? 14 is the superior mesenteric vein. So the names of the veins and the names of the arteries are the same. These are taking venous blood from the stomach, from the intestines, into the portal vein to take it to the liver, sinusoids, hepatic, absorption of nutrients, etc. You don't have to know all 18 of these, but you do have to know some of them, the ones that are on your key concept list. Let's stop. Is this the last slide I gave you?
Yeah.
Yes.
Yeah, so let's just finish this up in 2 minutes. This is water flow. So remember the way we did water in the kidney? Kidney is important for the reabsorption of water. The large intestine is important for the reabsorption of water as well. So we drink and eat a certain amount of water, and then we secrete a huge amount of water, 1.5 liters of spit, 2 liters of pancreatic juice and bile, 2 liters of gastric juice, 2 liters of pancreatic juice and bile. These are large volumes, 2 liters. Who's got a liter bottle here? Somebody's got a liter bottle. There's one. 2 of these of gastric juice and bile every day. 2 of these of stomach acid every day. So, and we don't excrete that much water, thankfully. We reabsorb most of it in the large intestine. So that's the job of the large intestine, is to absorb water. The small intestine's job is to absorb nutrients, and the large intestine's job is to really absorb water. I will not ask you to memorize the numbers here. So don't use your cognitive load to memorize these numbers. That's not a key concept. Okay, so we're done. Thursday will be a